Angelica Ramirez, and today I'll be presenting cell-mediated immunity. So what are the functions of helper T cells and cytotoxic T cells in cell-mediated immunity? Helper T cells secrete cytokines that recruit and activate other leukocytes to ingest and destroy microbes. Cytotoxic T cells, on the other hand, kill any infected cell containing microbial proteins in the cytosol and eliminate any cellular reservoirs of infection. What are the steps in the activation of T lymphocytes? The first step is antigen presentation. Antigens are presented on the surface of the antigen-presenting cells, or APCs, to major histocompatibility complex, or MHC molecules. MHC class I presents antigens to cytotoxic T cells, or CD8 cells, and MHC class II presents antigens to helper T cells, or CD4 cells. The second step is recognition by T cell receptor, or TCR. The TCR on the T lymphocyte surface binds to the MHC on the surface of the APC. The third step is co-stimulation. Co-stimulatory molecules like CD28 on the surface of T lymphocytes interact with the ligands on the surface of APCs, which works as the second signal needed for activation. The fourth step is activation. The T lymphocyte is activated through antigen recognition and co-stimulatory signals. The activation leads to gene expression and cellular proliferation. The fifth step is clonal expansion, which is the rapid increase in the number of antigen-specific T cells. Cytokine interleukin 2 stimulates proliferation. The sixth step is differentiation, which is the conversion of naive T cells to effector and memory cells. Effector T cells are in charge of eliminating microbes, while memory cells have a long life, circulate in the tissues, and are ready to respond to a secondary exposure to the same microbe. And here is a picture of kind of like what I just explained. What signals are needed to activate T lymphocytes? T cell activation needs both antigen recognition by TCR and co-stimulatory signals by interactions between T cells and APCs. The first signal is antigen recognition. TCRs recognize antigens when they are presented with MHC molecules on the surface of APCs. MHC molecules have class I and class II. Class I presents intracellular antigens to CD8 plus T cells, while class II presents extracellular antigens to CD4 T cells. The second signal is co-stimulation. T cells have CD28, while APCs have CD80 and CD86. The interaction between these molecules provide the necessary co-stimulation for T cell activation. What cellular receptors and ligands are used to sense and respond to activate T lymphocytes? The receptors needed are TCR, which recognizes MHC-associated peptide antigens, CD4 or CD8 co-receptors on the T cells that recognize MHC molecules on the APC and help the TCR to send signals. Adhesion molecules that strengthen the binding of T cells and APCs, and co-stimulators that bind to co-stimulatory receptors on naive T cells, promoting responses to pathogens. And the ligands are MHC class I and class II, and ICAM that are present on APCs. So this is a picture of the receptors and the ligands that are present on T lymphocytes. The receptors are on the left side and the ligands are on the right side. What are the types of T cell mediated immune responses? T cell mediated immune responses are designed to counteract distinct threats. CD8 T cells target and destroy infected cells, while CD4 promote the activation of other immune components and mediating communication between various immune cells. The regulatory T cells are very important to maintain equilibrium of the immune system, which prevent overactive immune responses and minimize autoimmune reactions. What are the subclass of CD4 plus and their role in cell-mediated immunity? The first one is T helper 1 cells, which stimulate phagocyte-mediated killing of ingested microbes and is induced by microbes that are ingested by and activate the phagocytes like macrophages. Their signature cytokine of T helper 1 cells is interferon gamma. T helper 2 cells stimulate B cells to produce antibodies, mainly immunoglobulin E, which helps in the defense against parasites. T helper 2 cells produce cytokines like interleukin 4, interleukin 5, and interleukin 13. T helper 17 cells defend against extracellular bacteria and fungi, and these produce cytokines such as interleukin 17 and interleukin 22. What are the mechanisms by which T cells activate macrophages? The first one is classical pathway, which is when T helper 1 cells release interferon gamma that activates macrophages to increase their capacity for microbicidal action. Alternative pathway is the second one, which is when T helper 2 cells stimulate macrophage activation, specifically antiparasitic responses by releasing cytokines like interleukin 4 and interleukin 13. What are the responses of macrophages that result in the killing of ingested microbes? By the activation of macrophages, their phagocytic and microbicidal properties are increased. Reactive oxygen species and reactive nitrogen species are produced by macrophages, and these damage microbial cellular components, leading to the microbe's death. Macrophages release lysosomal enzymes during the process of phagolysosome fusion, and these enzymes help break down internalized pathogens. How do CD8 and CTLs kill cells infected with viruses? CD8 T cells recognize an antigen on an infected tissue cell and releases granules that contain granzymes and perforin that enter the infected cells and induce apoptosis on the infected cell.